So the last talk of this technical session, who is an eminent person from industry, Dr. M.V. Nandakumar, who is Associate Research Director, Syngene International Limited, Biocon Park. And he did his MSc degree in MG University. And then he joined the group of Professor G. Vijay Nair in the Organic Chemistry Division of NIST Trivandrum. Then after completing his PhD, he moved to Germany and USA at Institute for Organic Chemistry, Technical University, Kloster, Germany, and NPG Research Institute, USA, for his postdoctoral studies. And he had bagged many awards and honors, and one of the remarkable one, on his name, there is a reaction called the Goldberg-Buchwald Nandakumar carbon nitrogen coupling reaction. So it's a big achievement to him. And he had published many papers in organic chemistry, and he had around 12 patents to his credit. With this brief introduction, I welcome Dr. Nandakumar to deliver the talk. Thank you.
It's not it, okay. Normally the last guys, we used to get a sleepy audience. So first of all, thank you, sir, for your kind introduction. And I would like to thank Professor Pradeep, sir, and Santosh Nandan, sir, and the chemistry department, head of the department, all the students for arranging this wonderful seminar. I never thought that this is this much big magnitude seminar, because normally you could see this kind of seminars in some IITs or ISCs, but in a college like Farook College, because I studied in UC College, Alway, I've not seen such a kind of seminar over. So you guys please continue this one, and we will support you at any point of time. Sir, thanks for calling me for this one. So, and I'm going to talk mainly for the students, not for the front row, because there are two reasons I came here. One is, we have a lot of opportunities for MSc students and PhD students, but we are not getting the students. I don't know the reason. Two reasons I can tell, because nowadays most of the colleges, they are not offering the MSc chemistry course, at least in Hyderabad. And second thing, those who are passing out from the colleges, the creamy layer goes to, of course, IAC or IIT and other places. We are not getting. But you could see that just like an IT boom in Hyderabad, now it's going to be a pharma boom. The reason is, you know that you have listened to about this COVID and post-COVID time. The COVID time, you could see the real power of the pharmaceutical industry, Indian pharmaceutical industry, because we were the one supplied essential drugs and life-saving drugs to the entire world. You can imagine that, if one of you infected in this room, maybe you can get a strip of paracetamol from a pharmacy. Suppose all of you are infected. Maybe the entire Calicut pharmacy can give you the drugs. But if the entire Calicut is infected, entire Kerala is infected, entire India is infected, entire world is infected, where do you get these drugs? There comes the importance of Indian pharmaceutical industry. Thank you, sir. We were the only one supporting the entire world for providing the cheap, I would say cheap, but high quality drugs. You see that every product in the market is based on the demand. For example, cars or anything, mobile phones. The producers will manufacture based on the demand. Same thing with pharma. But all of a sudden, within 1 month or 2 months, the drugs requirement has increased to multifold. So who can manufacture that much? There the answer comes, the Indian pharmaceutical industry. This is mainly because of our very strong pharmaceutical sector here, big manufacturing sector here, government initiatives, and of course, the skilled people like all of us. So with this introduction, I would like to start. Today, my intention is to just give some idea about what are the works we are doing in a pharmaceutical industry, mainly in the research and development. There, most of the students working are MSc chemistry students, M-pharmacy students. We have a lot of job opportunity over there. There are two reasons. One is the make in India. But we don't want to depend on China for any more raw materials or API. Second thing, there is a new act coming in US that is called Biosecure Act. Because the US is going to ban all the American companies to work with the Chinese companies. So eventually that all work will come to India. That is, I would say that is going to be another IT boom in Hyderabad, the same pharma boom is going to come. So today, I will just give you some idea what kind of work we are doing in an R&D by taking an example. So before going to that one, I will just try to give you some idea about what is a drug. For example, if you buy a Dolo tablet from a pharmacy, it contains, there are two components. One is an active pharmaceutical ingredient, another one is an inactive ingredient. An active pharmaceutical ingredient is a substance or an organic compound which is able to cure the disease or suppress or reduce your symptom of the disease. That is called an active pharmaceutical ingredient. That is called a drug substance. And this is an inactive ingredient because I am sure that all of you have made this paracetamol in the laboratory. Can you take that for a fever? Yes or no? No, we cannot take that one. This has to go through a set of experiments and this has to go through a series of quality tests, then only it is a drug. Because if you take sometimes this organic compound, it may be sour in taste, you may not be able to take that one. So here comes the formulation, here comes the synthesis. Today my talk is mainly based on this part, that is an active pharmaceutical ingredient. So I would say API in my discussions. And for example, in the paracetamol, say this is the active component, and this is called drug substance, and this is called a drug product. You can see these are the names available in the market. Crocin, Dolo, whatever you want to say, you can get it from. And you see that these drugs are available in the market just for maybe 10 rupees or 20 rupees. So suppose if the drug is very costly in the market, how many of you can afford that one? So that will kill the entire population. If the medicine is available, if it is not affordable, nothing but there is no medicine. So there comes the role of pharmaceutical industries and bulk products. So these slides, just I will give a brief idea. When a drug is discovered in the market, for example, the scientists from IACR or IITs or other places, they used to identify a new molecule and they will say that this is a suitable drug for such and such disease. Initially, they will work on some milligram scale. When they see that this drug is having some activity, they will go to clinical 1, clinical 2, phase 3, phase 4, something like that. And here they need more quantities of the material. Suppose if you are doing a 10 gram scale in the reaction, paracetamol synthesis in the lab, you can do it in an RB flask, one condenser, and say 80 degrees Celsius. But if you want to make it in say 1000 kg, because I have to give to the entire Kerala, I need 1000 kg of because 500 mg is the per dose, maximum 4 tablets. 2 grams per person is required if you are infected with fever. So it has to go through a different set. That is called process chemistry. Process chemistry is nothing but taking a process from a lab, from a milligram scale, into a plant, or a kilogram scale or ton scale. It should be cost effective, environment friendly, and reproducible. Then only this is a good medicine in the market. If it is very costly, nobody can afford that one. And it should meet all the quality parameters. And the drugs is one of the most regulated product in the world. Because quality, no compromise. If anything happened to the quality, it is just like a bus master. If you take that medicine, people may die. So this is the Indian pharmaceutical structure just for the MSc students. If you want to look for a job in the future, you can see that the Indian pharmaceutical companies are divided into innovator company. They used to introduce the molecule first time in the market, but these industries are very few in India. Maybe you can see that less than 10 because of the high cost and the time. And CDMO and drug discovery, whenever an innovator company identified a molecule, they need in kilograms or 50 kgs or 100 kg, they used to approach the CDMO companies. This is the company where I am working now. So we used to work closely with the innovator company, just like big pharmaceutical companies like Merck, Johnson & Johnson, Abbott, Pfizer. They used to ask us to make these compounds in good quality and in quantity. And most of the Indian companies, if you talk about all the Indian companies, are fall into this category that is called generic drugs. Generic drugs is nothing but a bulk manufacturers. They used to make it in kilograms or ton, even 1000 kgs, with high quality material. So I'm going to talk about the process which a molecule is taking from a laboratory to a plant and to a person. So this has to go through a different set of procedures, operations, and we need to follow a lot of rules and regulations because the final product is going to the patient. So government is there, so many authorities are there. They all put lot of restrictions, lot of specifications, and this is the specification quality is 100%. There is no compromise. You see, India, I would say that Indian companies are generic in nature. And because of this generic drug, the medicine is available in the market for a cheap price. Suppose if the branded company is selling a compound just for an antibiotic or anything, for an amoxicillin or a tablet, you have to spend maybe 1,000 rupees for one course. But when a generic drug is coming into the market, it will be less than 100 rupees. The reason is the generic enters after a patent protection of 20 years. And after that, the generic company has to do only this much analysis, this much test and manufacturing. But a branded company should do all these things. This is the animal studies and human studies. That is very, very, very costly. If something happened to a subject, the company has to pay millions of dollars for that one. So that's why the Indian generic companies can make the molecule for very, very cheap price. But in the drugs, this is there. Higher prices, higher quality, no. Even the generic drug is having a very low price, but quality should be higher. It is not like any car or any other thing. If you pay more price, it has a high quality. No. In drug, both the quality should be same. And when it comes to generic, we have a longer clinical safety data because the branded company is there in the market for almost 5 to 10 years. After that, the generic enters. When we enter into the market, we have all this safety data with us. So this is the basics of a generic and branded drug. But in all the cases, process chemistry play a critical role because we have to make that active pharmaceutical ingredient where the chemistry students and chemistry experts plays the role. So I would like to explain the process chemistry in a laboratory with one example. So I'm a process chemist. I'm a scientist, I'm a chemist, I'm an organic chemist. Again, I'm saying I'm a process chemist. When a process chemist is looking into a molecule, this is the one recently we developed in our laboratory. This is an anti-cancer compound, and it's called Jakafi. You can get this in the market also. It is used for bone marrow cancer, and also it is also used for atopic dermatitis. When I start a project in the laboratory, I should get some information before starting this compound in the laboratory. The first thing is, what is the activity? It's an anti-cancer compound. You know that if it's an anti-cancer compound, it kills the cancer cells, but it can also kill the normal cells. You have seen that people, those who have gone through the cancer treatment, they will lose the hair and they will be very thin because the normal cells also got degraded during this therapy. So I have to see, then the scientist working on this molecule, he has to take extra care. Otherwise, he will also get infected. So this is the information I'll get from this one. Then you see that the strength, you can see that in every tablet, 5 mg, 10 mg, 25 mg. And you can see the doctor will prescribe 40 mg only you can take maximum. This information also important for me as a process chemist because when I make this compound, this may contain some impurities like a carboxylic acid group or amide group during the process. Those impurities should be controlled into some level. If it is a 40 mg, I can have say 0.15% is the impurity with non-structure and 0.1% is an impurity with unknown structure. If it is a 2 gram, then I have to control the impurities below 0.05%. So this information I'll get from this one, and this is an oral administration. It's a tablet or injectable or a subcutaneous. For example, it's a tablet. It will go into your stomach. I'm talking about the human body, okay? It's nothing outside. If you are taking a tablet, it is going inside your stomach. And if there is some toxins, like if you rub your skin, some of the skins may peel into that one. That is an endotoxin. It can grow. But if it is going into a stomach, maybe it will kill because of the high acidic pH. But if the drug is an injectable, you are directly injecting into your blood. So that can grow inside your blood, and that is also toxic to your health. So this information I'll get from this one. Then you see it's a polymorphism. I think some of them are asking, the polymorphism is one of the most important parameter, physical parameter I have to set when I develop a molecule. Because you know that the sugar, there's a powdered sugar and there's a crystalline sugar. The solubility is different. If the innovator company, the first drug is in the market with a particular crystalline form, We have to make the same crystalline form throughout that lifetime. Otherwise, the activity may vary because of the solubility. And you see that this BET, then OEL band. Maybe when you are working in an organic chemistry laboratory, you are not worried about which band it follows. We have to check that in which band. This is 1, 2, 3, 4, 5, 6, 7. 1, 2, 3 band compounds, you can handle it in your common laboratory hoods. But when it goes to 4 and 5, these are called high potent active pharmaceutical ingredient. It has a side effect. These are all anti-cancer compound. So this should be handled in a containment facility, something like your COVID time containment facility. The chemist and the molecule should be separated from that one. So this many information I used to get before starting. All this information you can also get from these are all free information available on except the SciFinder reactions, all others are free information. If you want to check any medicine you are taking, if you want to get some idea what is the activity, what are the side effects, what are the dosage, chemistry structure, as a common man, you can get from an FDA label. Just type the ruxolitinib, any medicine name, type FDA label, you'll get a PDF. It is free. So, but don't treat yourself, only for information. So going back to process chemistry, this is the route which is coming out from a research laboratory like IAC or IACR or from any drug discovery company. They will do, first they want a compound and test it for an activity. They are not worried about the criticality, yield, time, cost, nothing. They want a compound as early as possible. So they want some 20 milligram or 1 gram of the compound. But when it comes to a bulk manufacturing, we cannot follow this one. For example, as a process chemist, when I look into this one, you can see that there is a sodium hydrate I am using. It is highly pyrophoric in nature. It can catch fire. There are multiple column chromatography. You see, if I want to do a 50 kg column, more than this room size column I need. That is not possible. And I need to use something like, say, 100 liters or 10,000 liters of solvent. That is also not possible. So I have to avoid all these complications in this one. And you see there is a chiral chromatography. And somebody was mentioning about thalidomide. You see, my product is an R isomer. But during synthesis, S isomer also formed. I have to remove that one, otherwise it will give some other side reaction. All these things a process chemist has to do. When I design a process chemistry route, you can see that I have simplified all these routes in a process chemistry by doing several reactions and looking into the literatures published by several universities and other institutes. You can see that I have avoided the sodium hydrate to sodium tertiary and I'm not isolating this called a telescopic reaction, and there is no column chromatography. I have just precipitated this product. And then I did a normal azamical reaction. It's a simple azamical reaction. Here they have used an expensive catalyst for that one. And I did, even though I'm getting a 1:1, 50% of the R and 50% of the S isomer, I have to lose that 50%. Still, my process is very simple and very cheap. So then I did a resolution technique using a dibenzoyl tartaric, and I have isolated, but you see that the EE is, the enantiomeric excess is 99.9%. And that is the requirement for a drug in the market. So I have simplified all these complications to a simple chemistry. For this one, we have to do a lot of experiments. That is called the process chemistry. I would just go through that process chemistry by explaining one step here. Just you see that in the first reaction, the protection of this nitrogen with a SEM chloride, and this nitrogen is protected. Then a Suzuki reaction with the boronic acid compound using palladium. This is the final compound are isolated. So when I isolate this reaction, when I fix this process chemistry, I have to make all the compounds, all the equivalents as minimum as possible, because this may be going for a 500 kg batch. One of the reactor batch, I may charge 500 kg of this compound. So even 0.1 equivalent can make a lot of money. So I have to optimize that equivalence first. There's 1:1 is the theoretical one. Maybe I have to use 0.01 equivalent extra. Toluene, the different solvent I have to screen, different temperature I have to screen, different time of the reaction I have to screen. So all this screening will be taken care by the synthetic R&D team. And a process engineers are there. Because when I set this process in the lab, it should be compatible to the plant. It's a bulk scale reaction. For example, one 500 liter or 1 kiloliter reactor may be this room size. So you can imagine the size of that one. In that one, maybe something around 3,000 or 4,000 liters of toluene, heating it to 80 degree. So you can imagine, and the scientist is standing near to that one. So if I don't do this process engineering and other thing, sometimes it may just blast that one. This happened into several pharmaceutical companies. Maybe 2 months back, you might have read in the newspaper, there is a company called Essentia in Visakhapatnam. There was a blast and almost 5 people died. Unfortunately, one of the scientists was my colleague. He was my reporter in another company. So if the process engineers are not doing the job, that is the end game. Why I'm telling this one is that when you are taking a tablet, please remember us, how many people, chemists are working behind this one. So another parameter which I told you already, I am mainly taking care of the oncology product development in my company. My team is mainly working on the onco product development because that is the high cost, low volume products. So most of the companies are doing cancer drugs, and you can see that nowadays the cancer is under control. Maybe 10, 15 years back, somebody is infected with the cancer, gone. But nowadays the treatments are there, more and more medicines are there. You can save a patient by taking a medicine, but the cancer compounds fall into this category. And this is my scientist, this is from our manufacturing unit. Whenever a chemist is working in an oncology facility, he has to be protected, and this is called isolator. All the chemistry we are doing inside the isolator to protect the chemist. Otherwise, you make the compound, chemist life will be in trouble. So, and this chemist has to go for a health checkup every 6 months. Even though this is 100% foolproof, there is no leak, but we will make sure that the chemist health that is our maximum priority. And the next one, so once you make the compound, nowadays the physical property is also very important. Maybe you may be asking about the crystalline nature. So earlier, there was no restriction like any polymorphism. Whatever the compound is coming, you can put it into market. Now, based on this one example, you see this is written over. This compound maybe you might have heard of during the COVID time. This was the first line treatment in the COVID. And the company where I was working, Heterodress, we were the #1 manufacturers in the world of this compound, all are antiretroviral agents that is against the HIV. And this is the way that at that time only India is called as the pharmacy of the world because we have supported the World Health Organization by providing these kinds of medicine to support the third world country. And you see that in this one, this product was manufactured by Abbott, and they put this in the market. Actually, it was a polymorphic form 1. And after some time, the doctors and patients were complaining that the activities are not as such what you declared. Then the scientists were investigating what happened. When they see that after some batches production, they could see that the polymorphic form, the form 1 was the marketed one. It was converted into form 2. And you see that solubilities of form 1 and form 2. Form 1 is 90 mg per ml, 19 mg per ml. So when you take a tablet, there is a dissolution rate, and there is a bioavailability of that one. So here you see that when a patient is taking, the dissolution is very, very low. So in turn, the medicine is not active. Instead of 90 gram, only 19 gram is active. So again, they withdrawn that drug from the market, again recount that one, and then finally they got the form 1. So it's the importance of a polymorphic form. And nowadays, every molecule we need, and more than synthesis, this polymorphism is the toughest job now we are facing. Because getting a stable form, it should be stable throughout the lifetime of the drug. It's nothing but when you purify a compound, you use ethyl acetate and hexane. One form will crystallize out. You use another solvent mixture, another form will act. And we have to make sure that this form is stable. So that is the most important parameter nowadays. And another parameter is we have to make the stability of this compound. We have to do some stress studies, whether this compound is stable in acid-base condition, or in the light, or in air. When you get a medicine from a medical shop, you can see they store it under protected away from light, and you can see that in an amber-colored form. You have to keep it, it should be tightly closed, keep it in a refrigerator. So we have to make all that information when you make this compound. So we have to expose it to high temperature and see this molecule is stable. And we have to see that acid base, what are the impurities forming, exposed to air, exposed to light. All this information as a process chemist, we have to develop. Because this is very, very important. Otherwise, whenever you get a medicine, after some time, it may not have the activity. Sometimes it may be poisonous. That is the most important. Activity, no problem. But if it is a poisonous thing, the pharma company has to pay millions. More than that, some people may die. That is really painful. So this is one thing, and you can see that, so please keep the medicine as per the prescription. If it is asked you to keep it in the refrigerator, keep in the refrigerator. If it is not, if they say keep away from light, keep away from light. Because if there is a conjugated double bond, it may isomerize. So please follow that one as a responsible chemist. And once I synthesize this compound, I have to test the purity of this compound. I'm going a little fast because only 5 minutes left for me. So there are some rules that is called ICH regulations. First, I have to check the organic impurity because it was formed during the synthesis. So many, for example, carboxylic acid, amide, all those impurities, that should be removed during my crystallization. I have to make it very pure compound. And then I'm using lot of inorganic compound like palladium acetate and everything. I have to control all the metals. Maybe you have given a nice talk. We are still using the silathiol and the cysteine. You can propose something to remove the palladium. Because the palladium, the limit is 10 ppm. And we used to do a lot of recrystallization. There are some method called silathiol. It's a polymer. We have to stir it with the silathiol or cysteine wash to remove the palladium. Mainly we are using palladium in the synthetic lab. And residual solvents, because we are using kiloliters of solvent, we need to control all these solvents. Polymorphism, I have already explained, particle size and BD, TD, these are some of the parameters a process chemist has to fix it. And then these are, I said, the organic impurities, and these are called genotoxic. And the most critical one, as I said, the other isomer. We should control this other isomer with a specified limit. Say, for example, below 0.15. I think somebody already shown this one, the thalidomide story. Because if this isomer, as per the regulation, I should control it below 0.15. If it is more than 0.15, I cannot sell because there is no data of that compound. So, for example, the thalidomide, I think this is a wonderful drug for the morning sickness for pregnant ladies. In 59s and 60s, 1960s, people took this medicine, the pregnant ladies took this medicine, but this was the result. The problem was this compound, the isomer is a teratogenic that entered into the fetus and killed, and this teratogenic activity happened. So nowadays, the governments are very, very strict on keeping the S isomer or other isomer component in the drug. And this is two examples, I would say, that the impurities, these two drugs, which our company were manufacturing. One is a sartan. It is a very common one. If anybody is having a BP in your home, they are taking this drug. It's a sartan drug for BP. And this is, I think, most of the people used to take for any acid reflux. Ranitidine, it is available in medical store. Both the drugs have been withdrawn from the market because of an impurity called NDMA, that is nitrosamine. So I am not going to the detail because the nitrosamine is that it will degrade in your stomach to a diazomethane, and this is a cancer-suspecting agent. So this is the solvents I already told you. You have to control the solvents. This is the solvents are divided into class 1, class 2, and class 3. Class 1 solvent, no. I'm sure that I think the new generation have not seen benzene. At least we have used benzene in our time, but the new generation, I'm sure it is not allowed, it is banned. And another one, already I said the inorganic impurities. And stability data, I already discussed. You have to generate the stability data, which temperature it is stable. And this also we have to provide to the formulator. And finally, after all this one, we have to make a specification like this. And in another 2 minutes, I just want to go. So we are not working on just paracetamol. How much complexity you can expect from a pharmaceutical industry? Nowadays, the pharmaceutical industry is doing extremely good job, very, very complex chemistry. If you want to come, you come. You can learn anything in the world, the latest development. So finally, I would say that this is the one I explained, from a drug, API to a drug. So this is one molecule my team has developed in last year. You can see there's a very complex molecule. There are 19 chiral centers. What is the number of isomers? MSc students, 2:19, 5 lakhs, 24,288 lakhs of isomers. You can calculate, I'm not telling any lie. So we have to make a single isomer from this one. And this is the total number of steps is 58. We made this one, and you have to, the specification is same for every molecule, 0.15% and 0.1% of the impurity with unknown structure, and 1 gram costs around 20 lakhs. This is a drug against the breast cancer. So this is already available in the market. It's called Havalin. It's an injection. And I don't want to go to the chemistry. These are the final 6 steps of that synthesis. And these are some of the other molecules which we are making. This is the remdesivir. I think everybody, all of you are aware of that one. It's a COVID time drug. The Hetero, our company, was the first one to manufacture this one and put it in the market. I've seen that the first time the people were putting milk on the photo of the owner, that is Hetero owner. Normally in Tamil Nadu, you can see that people put the milk on the film stars. But when we made this compound, this first time this happened in Hyderabad. So this is another compound for an excipient, and this is a peptide drug. And when you go for a MRI, the doctors used to inject one compound. That also we are making. And this peptide drug, as we mentioned, that there are 2 molecules are coming to the market. This will be the game changer for the diabetic patient. One is called semaglutide, and one is called tirzepatide. Those are all 39 amino acids. So we are making that compounds also in the pharma industry. And this is just, can I take another 5 minutes, sir? Shall I take another 5 minutes? So this is one of the latest developments, what we are working in the laboratory. It's called an antibody drug conjugate. As I mentioned, if you are taking a chemotherapy, you can see that the patient has to go through a very painful time. Loss of hair, weight loss, pain. Maybe sometimes the patient may think that better to die rather than going through this, because that much pain he has to go. The drug will kill the normal cells. This is a solution to that one. This is a oncology drug called exatecan. If you inject this one, that patient has to go through all these sufferings. But if you attach that one through an antibody, nothing but a simple antibody, I can say that the egg white. It contains some lysine molecule and amino acid molecule. You link this molecule through that ovalbumin through a linker. This is nothing but a dicarboxylic acid. Here it will form an amide, and here is an amide. And once you inject this compound into your vein, a cancer patient, it will pass through the blood. This will be inactive. And this ovalbumin or the antibody is designed in such a way that it will only recognize the cancer cell. Whenever this recognizes the cancer cell, it will be absorb the cell and encapsulate it and release this drug. So it selectively killed that cancer cell. That is called the biological missile for cancer therapy. So we have developed this molecule. Actually, it's developed by other compound. We have done the process, other companies. We have done the process development. These are all interdisciplinary research. We need chemists, biologists, analysts, engineers, polymorph scientists. It's an interdisciplinary science. So nowadays, the pharma companies are working together with all the people. So these are some of the equipments used in the laboratory. You can see all these kinds of analytical method, purification methods you will go through in the lab. And finally, I would like to dedicate this talk to my supervisor and my mentor, Dr. Vijay Nair. He is no more there, but because of him, I stand in front of you. And finally, I would like to thank Syngene and my boss for allowing me to come over here. So once again, thank you very much for your patience. Any questions?
Please, sir. Please, sir.
Extremely happy to hear this. Your presentation gave a feel for why one should be an organic chemist.
Thank you, sir.
And when I used to meet Vijay Nair, of course, we all knew his great organic chemistry prowess. He produced fantastic students. He used to put out every few years and accounts of chemical research and all that. But I see that people like you, he is directly contributing to the health of the country, world at large. Thank you.
Thank you so much, sir. Thank you.
Now, having asked this, having mentioned this much, I would say that touched upon the important point that in this emerging pharmaceuticals era, how we can be the leader. And that is possible only if academia put in the right kind of program so that they connect with industry very early. So maybe a few comments from you on that will be useful, because this is now the 4-year BS programs being built by the country in many institutions. Farook College is autonomous, you know that. So it is possible that these institutions can structure the program in such a way. The Vice Chancellor was here. So all those programs are possible. I was talking to him just the day before yesterday. I know that 4 lakh students are studying under Calicut University.
Mm-hmm.
And it is possible if we can structure it, it can contribute to India in a big way. That's all that I wanted to say at this point.
Yeah. Thank you, sir, for the comments. I just want to add 2 things, because you asked this question. So I really want the help from the scientists from IIT and IISc, because most of you are working on material science. We are facing a big problem in the industry from 2016 onwards. This drug, sartan, ranitidine, and there are several drugs. This has been withdrawn from the market because they found a contamination of NDMA. That's N-nitroso-dimethylamine. It is nothing but if you use any dialkylamine, triethylamine, diisopropylamine, diethylamine, and if you use some water, water contains some trace amount of sodium nitrate. This can produce this compound. And this is cancer suspecting compound. And the limit is 1 ppm, not 1 ppm. And after we make some 500 kg of the material, when it is analyzed, okay, this is 1 ppm, withdraw the material. And the company Hetero has withdrawn from the market. And this one company in Hyderabad, Saraka company, has shut down because of this content. So we really want help from the academic. A method to remove this one, not by a chemical reaction. Because if you do any chemical reaction, it will degrade my compound. If you come up with some silica or carbon treatment, we have to go through a filtration, because the cost we cannot add. You see, this is a BP drug. If I increase the price, you know that how many people are taking a BP tablet in India itself. They cannot afford. So really, this is one of the things I would say the academic people. Please think about it. You can get any information from the Google about this the nitrosamine issues, or you can contact us. We will give all information. Please spend some time to support the industry. And the second thing, as sir said, the collaborative works. So this molecule, sir, this is one of the best example of an academy and industry collaboration. This molecule is called halocantin analogs, isolated from C. And there is a big molecule actually. The first total synthesis has been reported by Professor Kishi from Harvard University. And he has given this to ESI company, and they found that this part is sufficient enough to do the activity. And they spend a lot of money to the university because of in the industry, we cannot spend too much time, 3-4 years for a compound. But academia, you guys are doing the real innovative chemistry. He has come up with the total synthesis of 58 steps and transferred into ESI. So there are still open for us. And as you sir mentioned, I think Rajiv you met already. He already contacted for some discussion. Of course, we expect some of the students come up with some basic idea of analytical research, regulatory knowledge, because all these molecules are going into the market. They should have some regulatory knowledge. At least in the MSc courses, if we can teach them what is HPLC, how to inject a sample, and how the impurities are separating, GC technique, just the basics, and how to analyze an NMR, not running the equipment, NMR, IR, UV, because we can directly take them. Now we are taking as a trainee chemist, 1 year we are giving the training to them, then only they are useful to us. If they come up with this knowledge, they can grasp very easily, and lot of jobs are available. Any other questions?
Yeah, I have. So very nice talk, very insightful. So You know that API, that which you talk about the API,
Yeah,
active pharmaceutical ingredient.
ingredient.
So sometimes what happens that you talk the amorphous to crystallization, polymorphs. So let's say certain that it is known, somebody crystallize it and one solvent, and that is good crystal and so the very better properties that you are telling crystallinity can change the that application and properties. So can we just that guy who can do the patent of that by making the polymorphs or the solvate of the API?
Yeah. See, whenever an innovator is putting a medicine into the market, you should have a patent on that one, 20 years. So in a drug discovery, it will take maybe 8 to 12 years. Maybe this is patent, another 8 years will be there. But as a generic company, like Indian companies like Hetero or Dr. Reddy's, or if you want to enter into market, we should not fall into that patent. So you might have heard about the evergreening of patent also. So what will sometimes in a one particular form only will give the biological activity. Sometimes other form can also give. So what we will do whenever, these are some legal like a NDA, NCA molecule, NDA and filing and everything. We could find out, we used to find out some solvates like salts, like say sulfuric sulfate salt or and this salt. If it is equally active like the original compound, we have to do a bioequivalence study. Then we'll go for that one. So that is possible. But initially we need to go with the innovator because he has developed that molecule. And sometimes what happened, there is a disappearing polymorphism is there. That means I got a particular form one time. It will never come in your another 100 experiments also.
But you cannot the patent that particular polymorph.
Yeah.
You have to go to the innovator only.
We have to go for a patent.
You can patent it?
Which one?
That you get the new polymorph. Let's say you are during the process, you have taken the API from the innovator, then you are doing that process chemistry. During that time, you find that there is some kind of polymorph, which is giving the better results or biologically more active. Can you patent that?
Yeah, of course. That is the game. I will patent it because I will say that my drug is better than the innovator. For example, I say different property, not only patent. You see, this compound, ESI has put it in the market. Sorry, PharmaMar or ESI. What they did, they have to purify this compound via preparative HPLC, chiral preparative HPLC. 1 gram is 32 lakhs per gram if I go for a preparative HPLC. Because you need a chiral column, chiral silica gel, almost in 40-45 lakhs for a preparative HPLC. Column of this room size, it is something another 1 crore kiloliters of solvent. What we did, we purified this by a simple crystallization. So where is flash chromatography and simple and the preparative HPLC, they got 97.5% of the purity. The innovator drug is in the market is 97%. When a process chemist like a generic company came, we made it is 99.8% purity by simple crystallization. We'll get the market. Even innovator will move out after some time. So it's a game. This is a money game. And in a patent also, like a new polymer, is just like a evergreening of patent. We'll patent it to block other people. You know, there is a thermodynamic form always come first. That's the most stable form. That somebody will patent it. When you do some other, it will come a kinetic form. And gradually it will move into the thermodynamic form. So these are all some other things to earn money.
You said that you get many products which are not useful.
Sorry?
You said that you sometimes get many products which is not useful when you scale up. So you must have a lot of waste. So how do you do waste treatment in...
I didn't get you.
How do you do waste treatment in pharmaceutical industry?
So the fundamental rule for a pharmaceutical chemistry is to minimum waste. Okay? But we cannot avoid waste. We have solid waste, aqueous waste, solvent waste. So this should be treated. There are some sewage water treatment plant in the manufacturing unit itself. We have to treat that water. We have to check that water, TDS and pH and everything. If it is pH neutral, TDS is within the limit, then only we can send that outside. Mainly we'll use for other purpose like, going for the toilet or going for the trees and something like that. But for the new batch, we should get always an RO purified water. For injectable type 1, we have to get a water for injection. There's very stringent limit are there. And solid waste and other waste will go for incineration. And solvent, there are some techniques, they will kill that with bacteria, some of the solvents. And some of the solvent, of course, it will be ignorant. Okay.
Just one general question. Usually the generic medicines are very cheap, comes under the heading generic medicines. So why the life-saving medicines are not available under, life-saving medicines, say for example, say something related to cancer, anti-cancer drugs, are they available under generic medicine?
Yes. There are two things. It's a life-saving and lifestyle. Lifestyle is all are available. It's a BP, diabetic, cholesterol. It's widely available. Generic versions are available. And if a generic is not available, I'm sure that Indian population maybe 30% is, because we cannot afford that one. Life-threatening disease, what you're talking is the cancer.
Life-saving medicine.
Life-saving medicines. All medicines are not available. If some of the medicine is under patent, it is available and the innovator can only sell. You might have heard about some treatment costs 5 lakhs rupees for one course. Only innovator is available because he is the only one in the market and he will fix the price because he has invested. One drug development cost minimum 1 billion, 6,000 crores.
Even after 20 years?
After 20 years, generic will come into picture. Then there are multiple suppliers. The prices will come down.
At present, there is no.
Sorry?
At present, there is no such kind of medicine which completed
Is there
20
almost
years.
out of 140 medicines for cancer? Nowadays, in every cancer, there is a medicine. Skin cancer, bone cancer, blood cancer, lung cancer, everything there is a medicine. Almost 55 drugs itself, hetero, the company where I worked, they are manufacturing under generic. And now the government initiative is there. They are also going to put so many medicines in the generics. But not all.
Yeah. Thank you.
So if you have no more questions, please join me, give me a big clap to them.
Thank you.
And you have presented the fundamentals of pharmaceutical chemistry as well as pharmaceutical industry in a very lucid manner. And being the organic chemist, actually I'm really enjoying your topic.
Thank you.
We are actually teaching lots of name reactions and nameless reactions, but we still don't know what's exactly acting in the pharmaceutical industry. So you have made it very wonderfully, sir.
Thank you.
So as a token of appreciation and our love, we would like to present a memento to you, sir. Please accept.